we have found that something is going on in the epigenome.
This is the most important step forward in our understanding of the causes of identical twinning in 100 years or so.
Tweeling zijn is het, denk ik, het meest leuke in het leven.
Identical twins intrigue, as they are a miracle of genetics. But impressive as they may be, they actually cause much confusion. Despite a century of amazing progress in most areas of science, we still have no idea how they arise. An international group of researchers led by Dutch professor Dorette Boomsma and young researcher Jenny van Dongen made a groundbreaking discovery. The researchers found a unique epigenetic profile in identical twins.
Basically, this has been a mystery. So identical twins, they are really mysterious subjects. They are like clones. They are genetically identical, and the way by which they arise and the reason for it has been a mystery. We really don't know why they split.
Well, we have studied mono and dizygotic twins in the Netherlands Twin Register for over 30 or maybe even 40 years. And the etiology of monozygotic twinning always remained a mystery. Some have labeled it random event. It's not random, it's very equal across time and space. But we saw in our research that genetics or environment do not offer an explanation. And then we had one tiny clue, and that was epigenetic mutations. Because in some imprinting disorders, there's an overrepresentation of monozygotic twins. And that tiny clue prompted us to study epigenetics in relation to monozygotic twinning, and then we had this enormous breakthrough.
But Van Dongen and colleagues went one step further back and asked whether the process of twin splitting left an epigenetic memory. Many of these differences were located in genes that help different kinds of cells recognize each other and stick together.
We were mainly interested in identifying whether epigenetic mechanisms are linked to monozygotic twinning. We also included dizygotic twins because we could compare them to the monozygotic twins. In addition, we also included non-twin individuals, so siblings and parents of twins. Here we all of a sudden saw methylation differences all over the genome. So when we analyzed all the methylation data from all of these twin cohorts, that at approximately 800 locations in the genome, there's a methylation difference that is present in identical twins when we compare them to other individuals.
Well, you know that family members often have a special bond, and identical twins are the most special pair of relatives because they have identical genomes. Their entire DNA sequence is the same, and all traits and characteristics in humans, like athletic ability, interest in sports, interest in music, being a good piano player, they all have a genetic component. So if you have identical genomes, you quite often have identical interests, and that already creates a reinforcing, very special bond.
Identical twins are the living proof of how genetics shapes our looks and traits. The researchers have found they carry a molecular signature on their DNA that no one else has. One that becomes fixed in their cells early in development and stays there forever. But what is this epigenetic signature exactly?
You can see the genome, well, you can compare it to this string. So basically within our cells, the DNA is wrapped up really tightly around small proteins called histones, and only those pieces of the DNA that have to be transcribed, they become unwound. It means that a very small chemical group called methyl, it's just a very small molecule, it's attached to the DNA, and together with other mechanisms, it influences whether other proteins can bind to the DNA to initiate gene expression. It also regulates how tightly this DNA is wrapped within our cells. You could compare epigenetic mechanisms to a piano. A piano player can play with the keys, playing them more fast or slowly. It's the same with epigenetic mechanisms. They can make sure that a gene is expressed very highly or lowly expressed, and they can also regulate when a gene is expressed.
The epigenetic music is played with the same notes at some parts of the genome in identical twins, and this is what distinguishes them from singletons and fraternal twins. One of the most famous identical twins are René and Willy van der Kerkhof, the only ones ever to appear in a World Cup final in 1978 in Argentina.
Dat zijn voor mij de mooiste momenten geweest in mijn leven. Dat we samen het allerhoogste in het voetbal gespeeld hebben. En dat is de waardering die je denk als tweeling wel hebt naar elkaar toe.
Ons moeder was alleen, we waren dan nog met 6 kinderen thuis. En ja, onze zussen die hebben onze opvoeding gedaan.
Dus we waren toch wel heel sterk afhankelijk van elkaar. En dat is misschien ook wel ons geluk geweest. Dat we dag in dag uit met elkaar bezig waren. Op school, voetballen, op het pleintje voetballen, achter het huis voetballen. Altijd met de voetbal bezig. En dan is het goed, denk ik, dat we met z'n tweeën waren. Want die kon samen niets doen. Iedereen keek naar ons. Want ja, we liepen, of we naar school gingen, dan liepen we altijd samen. Alle twee dezelfde kleren aan. Dus je kreeg altijd aandacht van iedereen. Nou, leuk.
If you had a car accident and you couldn't walk anymore, then you could not become a fantastic soccer player. So, you know, there's always an exception. But normally, the other half of a monozygotic twin that is a fantastic athlete will also be likely a good athlete.
We waren echt echte tweelingen. We waren echte tweelingen die echt gewoon in alles op elkaar lijken.
Je hebt een ander leven als iemand die alleen op de wereld is. We hebben altijd iets aan elkaar gehad. En dat maakt je denk ik ook sterker, ook bewuster en zelfverzekerder, omdat je met z'n tweeën bent. En soms kun je achter de ene verschuilen, aan de andere kant sta je voorop en dergelijke. Ik vind het een stuk meerwaarde hebben voor het leven om als tweeling te zijn.
How do identical twins differ from fraternal twins? Here are 2 pairs of twins. Peter and Sjoerd are identical twins, and Britt and Avi are fraternal twins. They will perform several tests in the laboratory. Identical twins show much more resemblance in balance and strength than fraternal twins. However, for some characteristics, such as flexibility, training becomes much more important during life, meaning that differences can and will occur between monozygotic twins during a lifetime.
Identical twins can be extremely similar. Of course, that is because they share all their genes. Dizygotic twins are like other brothers and sisters, but they happen to be born at the same time. They share on average 50% of their genes.
The processes leading to fraternal twins and identical twins are very different. Fraternal, or 2 eggs, or dizygotic twins arise after double ovulation, in which the genetic predisposition of the mother plays a major role. Identical, or 1 egg, or monozygotic twins arise from a single embryo that splits into 2 in the very early stages of pregnancy and retain the same base pair sequence of their genes.
An embryo splits, it's a huge event, right? And it's very well possible that this actually triggers a change in the cells that leads to these epigenetic changes, so that the signature that we find is actually an effect of the splitting of an embryo.
The twin split usually happens between day 2-3 and day 8-12 after conception. So before day 2 it is still one organism, and in very rare cases there can even be multiple splits. So we then get identical triplets, and there's even a famous case of 5 identical twin sisters who were born in Canada in 1934.
Among mammals, the human being is rare in terms of having monozygotic twins. There's only the armadillo is, as far as I'm aware, the only other mammal that has monozygotic twins. So it is a typical, not very frequent, but really human feature.
The other thing about monozygotic twins is that 10% have some congenital anomaly. So it's 3 to 4 times the background incidence. And again, from my standpoint, by studying monozygotic twins, we should gain insight into why these congenital anomalies happen and are they actually preventable.
You know, the exact rate of monozygotic twinning, actually, we do not know. Because one of the twins can stop in development and disappear during pregnancy. We call it vanishing twin. The co-twin that stop in development can influence in a negative way on the survived twin.
A special case of this phenomenon has come to visit us at the laboratory of the Netherlands Twin Register. This is Lucas. His twin brother stopped developing in the womb, so Lucas was born alone. He will mainly test his heart today in the lab. And he is also curious about his vanishing twin brother.
Is er ook iets met ziektes, of misschien ook juist de goede kant, iets met talent? Heeft dat ook te maken dan met een-eigen tweelingen of vanishing twins door die verschillende epigenetica?
Ja, men heeft die vraag inderdaad gesteld bij allerlei patiëntengroepen. Er zijn aandoeningen, nou bijvoorbeeld een bekende aandoening waar die vraag gesteld is multiple sclerose. Ik heb een-eiige tweelingparen waarbij de een de ziekte heeft en de ander niet. Kan dat verklaard worden uit hun verschillende epigenetische profielen? Dat heeft tot nu toe nog niet zo heel veel opgeleverd.
Ik ben hier vandaag inderdaad op de VU, maar het gaat eigenlijk niet om mij, maar om mijn zoon die achter mij zit, Lucas. Die een van een eigen tweeling was, waarvan zijn, nou, omdat het een-eiig is, kan ik zeggen broer, maar dat is misschien iets te groot gezegd. In ieder geval de andere waarvan het hart nooit is uitgeklapt, maar een lange tijd heeft meegegroeid op zijn pomp. En gelukkig moet ik zeggen, het is een beetje gek om dat zo te noemen, maar is dat na een week of 15 is de bloedstroom daar naartoe die volledig op gang werd gebracht door Lucas, is langzaam dichtgeslipt, waardoor hij zelf vrij door kon groeien, omdat je dat met een klein pompje van wat één hart eigenlijk niet kan doorzetten in je eentje om dat allemaal mee te laten groeien. En bovendien zou dat ook geen levensvatbaar kind zijn geworden.
Lucas will undergo several tests to check his heart function and stress response. Lucas is lucky. He doesn't have a congenital anomaly as a consequence of his vanishing twin brother. But there are many opposite cases, and such people might not know that it's because they had a twin. This could be detected thanks to the new discovery of epigenetic signature.
The recent publication by Jenny van Dongen from Dorrit Boomsma's group is highly relevant because that study showed that persons that are part of a monozygotic twin bear a genetic signature that were imprinted at time of development as they were the embryo, will be able to distinguish those that are indeed part of an MZ twin and those that bear the signature but are singleton. So that will tell you the rate of MZ twinning loss.
Part of what we're really interested in with the epigenetic work is that we can identify an individual who started as a twin, because most of the children born with these very specific type of malformations are actually singletons. Now, from my standpoint, that would fit perfectly well with that one twin dies and the other one survives.
There are many potential applications for epigenetic signature for monozygotic twins, and one of them is in clinical. For example, some families want to know if they have twins running in the family, if there are previous history of monozygotic twins, so we can know after the birth and if there are twins that are running in the family. So it could be interesting also from a family perspective, from scientific perspective, from research perspective.
Well, there are multiple routes into the future. Very fundamentally, we hope it will give new insights into embryology, developmental biology, and that the colleagues who work in those fields will pick up this finding.
Why bother to put men on the moon argument? I mean, putting men on the moon has got no intrinsic value whatever. But in the course of doing so, it opened up huge new areas of technology, from things that are extremely complicated to simple things like the Teflon fry pan, that we all use and enjoy. And all of these were spin-offs from from that research. So I very much hope that the work we're going to do will seed some successes like that.
So all in all, I'm a great advocate of a much better understanding of the biology of monozygotic twinning. But nowadays we apply technology in assisted reproduction that causes an increase of monozygotic twinning, which makes it much more important to really understand why monozygotic twinning occurs.
There's a huge interest in this finding from the general public. Many people suspect, or they know because they have heard this from their mother, for example, that very early in pregnancy there has been a twin pregnancy and that they lost their twin brother or sister. And many of those people really are looking for some sort of confirmation. And what we show in our paper, of course, is that this signature is so stable across life that you can, so to speak, look back. Is it likely that I once in my development had an identical twin brother or identical twin sister?
Of course, the dream would be to find out what is it exactly? What is the cause of identical twinning? We now have made a very important first step. We have found that something is going on in the epigenome, but we don't know exactly when. We don't know exactly whether it's the cause or the effect of the identical twinning event.
Monozygotic twins can help us to understand in what manner embryos develop, how assisted reproduction technologies and infertility treatments influence the embryo, how some of congenital disorders appear during the prenatal period, and how the vanishing twin syndrome affects one's health. The epigenetic signature contained in the DNA of monozygotic twins is a type of music in the genome of which only monozygotic twins know the notes. And from now, scientists can read this signature, the key to the mystery of monozygotic twins.